thank you. You know, if there are any Monty Python fans, this would be the good time for the foot to come down. And now for something totally different. You know, this project grew out of an idea about 20 years ago when it was clear the omics revolution was just going to transform environmental sciences. And the question I kind of started to grapple with was, given all that information, how could you turn that into a sensory tool, you know, a remote sensing system? And that's what gave rise to this idea of the ESP. Many people have been involved with it, a lot of sponsors, and I'll leave it to that. For the purposes of today, if I can get this to work. So I want to pose this idea of what is an ecogenomic sensor? How can we get this phrase into the lexicon of the genomics, environmental genomics? I'll give you an example. The ESP is just one embodiment of such a device, and leave you with some food for thought on how it might evolve in the future. And interestingly, this image came out from the Center for Environmental Visualization, University of Washington, not far from here. And it captured and kind of codified this notion of ecogenomic sensor because they use that phrase to describe it. And what the notion was is that you had this device, totally ill-defined, but it was pulling in all kind of manner of microorganisms and spewing out a kind of genomic view of the world. But what's important about this image, and for those of us who are working on trying to realize, you know, to put a face on this device, was that first of all it included lots of things. Secondly, it's embedded in a much larger observing system. The data itself is meaningless without the rest of the environmental context. And third, it spoke to scale. We have sort of the sense of ocean color. So networks of systems embedded within systems in order to really make sense of this information. And so the work we were doing was really very fundamental in you know, how do you actually come up and build such a machine? And it is very simple conceptually that it begins with sample acquisition. Some of the things we're after are pretty dilute. You need large volumes, and that's not trivial. It's very different from the biomedical, you know, sort of single cell type analyses. A way to concentrate those targets, analyze them, and then publish all of that, essentially doing cloud computing. You're putting it out, and the clients that are interacting with these devices from afar. It is totally hands-off. And so with that, I offer you the definition of an ecogenomic sensor. It's really simply something that is allowing us to apply molecular analytical techniques totally outside of a laboratory. But what's important is it's integrating everything, sample acquisition, processing, and data transmission with no human in the loop. And that's very much in contrast to what most of us, I think, practice in this field, which I will call ecogenomic sensing, which is really the same thing, except you require a human at some step to move a sample or press a button or whatever it has to be. And so here is this embodiment of an ecogenomic sensor. We didn't call it that when we started, but it's become that. We've adopted that phrase thanks to Hunter Hathaway at Center Environmental Visualization. It's known as, we have it sort of in a modular device, which is the core. This is the thing that's kind of in that part of handling the samples and processing them. Downstream, there are what we call analytical modules. These are devices. If you think about the handheld devices for biomedical research, for example, this would fit very well here. They do lots of things, but they want some upstream handling and processing, and they like to live in a say, a one atmosphere housing or something. And then outside of this, if you get to situations that are extreme, eventually you just run out of runway. The core cannot handle all environments under all circumstances. At some point, if you want to collect a sample, you're better off to use some other device. And so what we decided was the way these things could work is if we defined interface standards for power, fluidic, and communications protocols, it would be like a USB port on your laptop. It would allow it to do lots of things. And so this device, as it stands currently, really rallies around a few core functions. The first is using these low-density DNA and protein probe arrays. I'll just show you a little example of it. Very simple chemistry for real-time analyses, you know, on 12 volts underwater. Same thing with quantitative PCR. Ability to collect material, purify the nucleic acids, run assays absent any human intervention. And last but not least, sample archival, which I think is critical because there's so much out there we don't know. We just heard about this. And there's always other chemistries that you would like to apply that just really aren't practical by today's standards to put in the environment and to make completely autonomous. And so we wanted the ability to bring material back, and not to mention that interesting things always happens on nights and weekends and holidays when you're not around. And so you'd like to be able to have some way of going back to verify these real-time results. So this interface to the back end is important because so many of the devices that are out there now are all running on these microfluidic scale or even lower. And so our system really on the front end is dealing with many liters of sample, primary raw sample. It's rendering it maybe to mil or less, hundreds of microliters in terms of some processed material. But when we want to get down to the manipulation, it needs something else. So we created a device that interfaces to that core, which can operate all by itself, standalone, or by a single fluidic and electrical connection, brings it onto that core and is now deployable. And so we focused first on a module that would allow us to do DNA purification and quantitative PCR, and it comes out of, ironically enough, out of home security land, Lawrence Livermore National Labs working on autonomous pathogen detection systems. And so this, you may not be able to see it well, but this small little black box is a 2-channel real-time PCR module. It's about the size of your fist. And there's a reusable solid phase extraction column. We use coils of tubing to put our reagents. We've shown for 10 months we can stabilize PCR reagents, for example, room temperature, and even some extremes on either side. So this just gives us a way to kind of develop these systems, proliferate them, and then deploy them in the ocean or in the environment. So working on this interface control standard, there's a whole bunch of other modules that I don't have time to go through today, but things which are far more capable and more rapid than we have deployed to date. So here, I'm not going to really talk about how this machine works. I just want to give you some example data. These are ribotyping arrays. You probably can't read it. There's a bunch of garden variety microbes from marine systems. This is about 10 millimeters on the side. And these rows are different targets, different ribotypes. This is a direct detection of ribosomal RNA from a crude homogenate. Sample 1, sample 2. The patterns are different. And if you look here, you would see these bugs, group 1 Archaea, are present in this case, and then they seem to disappear from the ribotyping reference. And from that, we can run quantitative PCR. A split of that same sample is parsed, run on quantitative PCR for a number of targets, so for time 1, time 2, and the target is shifting in its abundance. So the DNA is detectable, but not the RNA directly. That's just an example of some of how we run. So this is occurring in real time, subsurface if desired, and using this kind of cloud, so to speak, to broadcast its data. A bunch of platforms on which these devices have been deployed to date. And I'm just going to mention a little bit of work from this, some near-surface moorings and deep sea, Drifter and Pier. A lot of this has been driven by this interest in understanding biogeochemical cycles and kind of picking off these key genes that are modulating these transformations of various substances. And a lot of this work is done with this Seymour Center in University of Hawaii. And you get a picture that looks a bit like this. This is from a mooring, for example. There is a whole variety of data: wind forcing, chlorophyll, nitrate, temperature, salinity, and then the abundance of different genes and the various ribotypes in the community of these organisms over time. It's kind of a 2D slice as water's passing by this device. And with this, you can begin to start to see how these organisms that we detect relate to, in this case, nitrate, salinity, temperature. And for these group 1 Archaea, which tend to be found in deep waters, they're almost like a positive control. When it's cold, salty, and very nutrient-rich, you tend to find them, and it goes down along the line until you reach a place where you no longer see them. It's not the environment in which they live. The point about this is that we now, with with this kind of perspective on these different communities, it allows us to begin to program the instrument to respond to the environment with which it's in. And you can ask questions about, are these bugs present under certain conditions, through an autonomous response. Not a human in the loop saying go, or at 8 o'clock in the morning turn on. The instrument responds to the environment itself, informed by this kind of cloud of other sensors in the area. Okay, I just thought it would be fun to show you what happens when you go deep. This is a device. It's the same exact machine, but it's now sitting in this titanium sphere, and that's rated for 4,000 meter applications. And we took it just this past summer, not far from here. Here we are in Seattle, and just south and west is a subsea volcano known as Axial Seamount, and it's part of a wired system that the NSF, through the Ocean Observatories Initiative, is creating a cable system that will allow you to plug instruments in, literally plug in underwater, to a network and give you power and data comms. And so we were exploring the use of the ESP in this site. And here it is. If you could see it, it's sitting on the bottom. This is at 1,600 meters. Here's one of those plugs that you use to get in and out of the network. And at a distance, we are in, this is a hydrothermal vent field. We're in the caldera of a volcano. And so we had a sample wand that ran out, and we put this little tip down into one of the cracks where the hot water's emanating. And this thing was running for a while. I'll just give you an example of some of the data. If you sample the ambient water around you in the caldera, the background seawater, it would be very cold, 2.5 degrees C, and salty. And as soon as you switch that and point that wand at the vent fluids, it's hot. This is actually not extreme hot. It's warm. It's less salty. If you look at the ribotypes associated with those fluids, very different ribotypes. It's obvious just by your eye. And then certain genes that we were after using quantitative PCR to show. So the point of this is really to say that even at 1,600 meters below the ocean surface, you can begin to think about doing interactive experiments using molecular biology. Okay, so this is a fun example of what happens after we build a lot of this for environmental, if we're looking at basic research, is how can you use this tool now for a more practical application, shall we say. And this is with Center for Ocean Solutions at Stanford University. We put one of these devices out on the Santa Cruz Wharf. It's just down in Monterey Bay. And the instrument is sitting here in this can. This is a utility closet. The main deck of the pier is above this fellow Roman Marin. The floorboards, if you see those gaps, occasionally waves will come and water splashes up underneath. And here's actually one of those sampling modules. It's a way to just pump water to the system, and then it drains back out through the floorboards. And we used a water feature, like a fountain for your yard, is what's pumping the water. And very hard to see here, but there's a radio antenna that's pointing to our software engineer's house, which is a little ways on the other side of the beach, and he is our gateway into the web. And so here's just an example of some of the data from this. We're looking for fecal indicator bacteria and human source indicators. Rainfall, there's an action level for EPA standard. What's important about that is that really we were waiting for rainfall to drive the source indicators up. Didn't happen until there's no rain, and we get this huge spike in Enterococci with a human origin. So totally disconnected from rainfall. I think this is the first example of an autonomous detection of sewage indicators that's ever been done anywhere. So clearly there's lots of room to improve on this and drive it further. Okay, so what's happening right now is we've deployed networks of these instruments in small locations, but for the first time ever, we're going to do what we call a global deployment. We'll have 3 sites, shellfish farm in New Zealand, which is subject to both problems with harmful algal blooms as well as freshwater runoffs and human pathogens and so on. Right here, Puget Sound at Friday Harbor, and then on the East Coast in Gulf of Maine. And these instruments will run simultaneously. They'll run many of the same assays, and they'll all be available from a GUI. So anywhere you have a wireless connection, you would be able to see these machines, get data, see what their schedule is, and alter their response to the environment as you see fit. And I don't think anything has ever been attempted like that, so we're anxious and excited about that. Okay, I want to end on this problem of time and space, which has, I think, become to us one of the bigger challenges in microbial environmental genomics, and that is that typically when we collect samples for that type of analysis, we do everything we can to reference the genomes, their transcriptomes, proteomes, whatever's going on to that instant in time, to the moment that we collected the sample, yet the biology is all a product of the past. It's a whole series of events that have led up to whatever manifestation that you have at that instant. And so the question is, how can we gain a sense of the history of these populations in order to understand what may have given rise to a particular group of microbes, population, or what have you. And so to do that, we took the same instrument, we put it on a drifter and deployed it in the ocean and let it drift freely for days. And just as a sort of an example, this is Monterey Bay, San Francisco Bay. It was a ship track that ran out. We came back and deposited the ESP here, and we just let it drift along. And as it's drifting, we used autonomous vehicles that would circle around it to ascribe a volume, to give us this kind of contextual volume as it moves through space and time. And just a snippet of this data from a work that is being done at Ed DeLong's lab with Elizabeth Otteson, one of his postdocs, is if you look at where we collected, these are samples that have been collected and just archived in this particular case, and have then done transcriptome sequencing on these samples and assigned taxa based on those reads to look at how they evolve over the space and time scale, and you start to see something like this. So in this case, it's just a Synechococcus reference. We see the strong circadian rhythms in clock genes, in respiration, and in carbon photosynthesis type genes. And so this is both time and space as the microbes are evolving. And I think it's a first for Ed DeLong and Liz Otteson pull out these patterns from this transcriptome, metatranscriptome, and there's also a whole lot of interesting things that they're seeing in how there's this alteration between transport functions and then other cellular metabolisms as the cells kind of bounce back and forth, at times anticipating changes in the photoperiod, for example, or day length. So very interesting. But that's all drifting. You're not really Lagrangian, and so we are focusing now on a system that allows us to do this autonomously. It involves a reconnaissance tool, the use of these vehicles that are very long duration, speedy vehicles. They can also stop and float and drift for surveillance, which gives us a way to track patches in nature, and then with that, target special ones for other vehicles, other classes of vehicles that do sampling and analysis. This is happening at Mbari. And this is where I think it's all headed. Is currently we've been using this device as a kind of model to understand how systems work. We're developing new mobile systems. This is about the size of a basketball, the sensor system that would carry out those types of measurements. And in the future, long duration platforms like profiling floats and others are certainly possible. And so I always love to use this slide for a final remark. You know, here's Wilbur flying, and I imagine Orville standing there thrilled. You know, they're going to get 100 yards in 30 seconds in the air, or whatever it might be. And someone next to him says, that's really cool, but what about personal in-flight entertainment systems and, you know, transcontinental flight? And, you know, and you can imagine Orville's response. At any rate, so I think this is it's a good metaphor for where we are with ecogenomic sensors. You know, we measure our success in months of deployment, you know, in dozens of assays, and we're getting there. But importantly, though, I think it's the concept we have demonstrated is valid. And I think that with all the Earth and ocean observing system infrastructure that's being put in place, we have a really unique opportunity to leverage this kind of device and to carry out environmental sensing in ways that have never been done before. And in particular, from a molecular analytical perspective, we now can begin to pose questions and answer them without necessarily being in the field. that's a real paradigm shift, I think, for environmental science. In particular, data visualization, how you incorporate such density of this genomic data, omics data, in the context of the environment and see it in space and time. It's a huge challenge. So that's all I have to say. Hopefully I got us a few minutes back on the time, and thank you for your attention.